Hi, all. This is Kermit the Frog.
Actually, it's Mr. Gonzalez. We are going to do our DNA notes. DNA. And I'm going to try to go a little slower this time because last time when I raced through the movie, the sound and the writing was just a little bit off. So I'm going to try to go nice and pace to see if it works out. I actually talked for about 2 minutes just now and I realized I didn't record anything, so I was talking to myself. That was just awesome. So here we go. Let's start over. DNA. We're just going to do DNA notes, and then in other lessons we'll do touch on the other stuff. So DNA, the first thing is, where is it? Where? Well, first thing, we'll get an organism. There's an organism. Wait. There's an organism. And what we're going to do is we're going to take a cell. Take a cell. There's a cell. And where do you go next? This is cell. We're going to go in the nucleus. Go there. And then grab the next thing, which will be what? Hot dog? No. Chromosome. There's a chromosome here. And then after that, we're going to grab this and look at it really close up, and that's where you find DNA. Because this right here is chromosome. This right here is DNA. This is a gene. And a gene is a piece of DNA that codes for a specific trait. Okay? Now, we also learned a lot later that this is actually this. Whoa. Val. Pro. Would vowel and illy. These are an amino acid chain right here, and this codes for proteins. Amino acids are the building blocks for proteins, and proteins are what makes your traits. So this code, this DNA code up here, this DNA double helix, that's the shape of it. It codes for a specific trait, and that this amino acids give you specific proteins. Now this right here will eventually, the amino acids make this awesome, cool 3D protein. And one thing that's important is this shape, because the sequence of bases in DNA gives the protein a specific shape. And shape is related to function. If you change the amino acids, you change the shape, you change the function. Okay? Cool. Let's go to the next. Actually, let me see if I just erase this. If I erase this, will it be better for the movie? I'm going to try that. All right, next word, genome. Genome is all the genetic material in an organism. In an organism. Now, it's not necessarily all the genes in an organism. You could have a humongous genome and just a few genes. Okay? Or you can have a tiny genome, like that, and have a tiny genome and have lots of genes in there that take up the whole thing. Okay, so genome is all the genetic material in an organism. Let's see if this is delayed now. Now, what did we talk about? Well, we showed you some different, in class we talked about different organisms and their genome. We said that amoeba versus humans. We actually said that there's a species of amoeba that has a lot more genome, a lot more genetic material, larger genome. And even if you're talking about genes, we said something like humans have around 30,000 genes and some amoeba have 4 million genes. But just their genome is larger. Now, what was the point of talking about this? The point was this, that amount or number of genes or amount of the genome or amount of genetic material is not related to organism complexity. And what that means is you don't have to have a lot of genes to be special. Amoeba are special too. Amount of genetic material doesn't relate. That makes no sense. There's no correlation. There's no correlation between genetic material and complexity. No correlation between genetic material and complexity. What does it mean to be complex? Well, have lots of different types of cells that do all kinds of cool things. So it doesn't matter. Amoeba have tons more DNA than humans, and they're just little blobs. Okay? Cool. That's the second point we made. Now let's talk about the DNA molecule itself. Again, we drew it like this, which is the double helix famous shape. 1, 2, 3, 4. We always draw like 4 lines that are in there like that. And the first thing we wanted to look at was, what is each little piece of DNA made up of? And each building block of DNA is called a nucleotide. A nucleotide is made up of 3 things. 1, a sugar. 2, a phosphate. 3, a nitrogenous base. Okay, those are the 3 building blocks of a nucleotide. And nucleotide is the building block of DNA. You need a lot of nucleotides to make your double helix. And you need these 3 things to make up this. These 3 things, 1, 2, 3, make up the building block, the basic unit of DNA. Do you understand? Okay, so let's try to explain what these 3 things look like. Now, when you get to chemistry, you'll learn these little symbols a little bit better, but this is a sugar, which has 5 carbons, and the name of it, awesomely, is deoxyribose. Deoxyribose. That's the sugar. That's number 1. Check. Phosphate. The phosphate is over here. It's bonded to the sugar, and we'll draw it as a little ball over here. That's the phosphate. Check. And last, the nitrogenous base. We'll draw as a little flag over here, and we'll put a letter in there. What letter do we want? I don't know. We'll put the letter A, because that's what we want your grade to be. This is the base. And there's 4 types of nitrogenous bases. You know them as A, D, C, and G. Those are the 4 bases. They stand for, now for the test you don't need to remember their names, but you should recognize if you see one. For example, on a multiple choice, I probably won't use A, T, C, G. I'll use their names, but obviously you know what they are by the first letter that they stand for. So if these are the 4 choices, you won't see them as single letters. You'll see them as adenine, thymine, cytosine, and guanine. Okay, but just know that they are, of course, are ATCG like we've been using in class. Now, just like we learned in class, A and T hook up together to make the characteristic ladder rungs on here. So here you would have an A, and here you would have a T hooked up together. And C and G hook up together. Okay? Now, what sort of things will I ask you? Well, the first thing is you're going to need to know, because DNA, if we unwind it, is 2 sides like this. I will give you one side of DNA. You will have to give me the other side. So if I put cat, gaga, I will ask for what's called the complementary base. And that just means the other side. So, you know how to do it. The first one, cat. What goes with C? G. What goes with A? T. And so on. You just fill in the letters. A always goes with T. C always goes with G. And so this would be my answer right here. Now, the next thing we talked about is that is awesome because that's also how DNA copies itself. We said that DNA, to replicate, unwinds itself like this. Okay, and what it does is, and on one side I'll put cat, and on the other side, which opened up, I'll put what goes with cat. G, T, A. Guitar. Now, inside the cell, there's little bases that float around. So you got your little, over here, you got your G's and your C's and your T's and your A's and your A's and your T's and so on. And they will go hook up with their partner. So over here, you'll get guitar. And over here, you'll get cat. And they make a new strand. Like that. So what you end up with after replication is 2 new strands, each with an original template. One side is from the original DNA, one side is new. Okay? So that's DNA replication. Next. What's last? RNA. We'll just do RNA really quick. And RNA is totally the same as DNA, except it's got a couple of differences. Number one, it's single-stranded. 2, RNA has no T. Not. It has U. Aw, it has U. This stands for uracil, in case you need to know that. And blah, 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 blah. Okay, and the sugar is not deoxyribose. The sugar in RNA is ribose. Those are the 3 differences. Okay, so that's DNA versus RNA. And I think I'm going to stop there because I want to check to see if the movie worked okay, and then we'll see if we, if it worked and the timing worked and stuff. So I'm going to double check it, and I'm just going to